All right, class. So picking up on where we left off with looking at the actual structure of the internal components of our muscle cells, we can now utilize that information to build an understanding of how those sarcomeres end up contracting. So how do we get from nervous signal, conscious control, telling our muscle to move, to the actual contraction of that muscle, okay? So we're going to be looking at the neuromuscular junction, which is describing where the neuron meets the sarcolemma and passes on that nervous signal to create an action potential on the sarcolemma of the muscle cell. And then we'll look at the actual mechanism of contraction. So these are two separate ideas. The neuromuscular junction is a location that's where we are transferring the signal from a neuron to the muscle. And the mechanism of contraction is the response to that signal once it actually gets to the muscle cell. So a neuromuscular junction looks like this. You have a motor neuron with multiple synapses, multiple of these axon terminals, these tendrils, all along the sarcolemma of the muscle. And that is creating a motor end plate. So where the motor neuron meets the muscle cell, all of its connections like that, all of the synapses together create one motor end plate. And then if we zoom in into each specific location of where a synaptic terminal is meeting the sarcolemma, that is our neuromuscular junction. So here's those definitions, I guess, of kind of what I was just talking about, right? So I'm gonna go ahead and move through this. I don't think there's anything I need to really expand on that's more than you just reading definitions. So I'm gonna move into the actual process. So again, as I was describing in the last portion of these recordings, okay, the neuron is going to have an action potential traveling down it that reaches these synapses. We then want to transfer that action potential onto the sarcolemma of the muscle cell so that it can conduct that signal down these T tubules into the cytoplasm of the muscle cell, the sarcoplasm, where it will then stimulate the synaptic terminals of the sarcoplasmic reticulum. Those synaptic terminals are carrying vesicles full of calcium. And so as a reaction to that action potential, the synaptic terminals will release that calcium, which is going to move some of those protein fibers on the thin filament, which now allows for the thick filament to grab the thin filaments and pull them towards the center. And that is what produces tension, produces that contraction. So starting with looking at our neuromuscular junction, what is happening at the location where the neuron's synapse is reaching, or not touching, there's space between it, but reaching the muscle cell, the sarcolemma. So an action potential is traveling down that axon to the synaptic terminal. As it reaches the synaptic terminal, that is going to cause the release of a neurotransmitter called acetylcholine. This is going to be a really important neurotransmitter that shows up in our later chapters too, talking more about nervous signaling in Unit 3. But acetylcholine here is playing a big role in contraction of muscles. So the action potential arrives, that stimulates the release of acetylcholine. Acetylcholine is then going to bind with these special receptor proteins that recognize that neurotransmitter. So as acetylcholine is binding with those receptor proteins, that is going to open some sodium channels, allowing for sodium to flood in to the sarcolemma, the sarcoplasm across the sarcolemma. And essentially the movement of ions like sodium is what generates these electrical signals, these action potentials. And we're going to expand a lot more on that in the next chapter when we get into Unit 3. But for now, recognizing that sodium moves into the cell after acetylcholine binds, and that is what causes an action potential along the sarcolemma. So now that action potential, this electrical signal, is going to travel away from this neuromuscular junction and down to those T tubules. So I'm moving a bit too fast there. So away from the neuromuscular junction, where it will continue down the T tubules. And then now we need to clean up, essentially, to stop that signal from just continuously going, or else you'd get stuck in a contracted state. And so we have this other protein that is there to clean up that acetylcholine. Because if acetylcholine stays bonded to the receptor, then those sodium channels are going to stay open. And that's just going to cause a continuous action potential to keep going, keep going, keep going. So acetylcholine esterase is this protein that is there to clean up that acetylcholine, bring it back to the neuron, and now we're back at our initial resting state. And then if we're going to send another signal, the process repeats. Okay? So that is the neuromuscular junction. Action potential arrives, acetylcholine is released, acetylcholine binds with receptor proteins on the sarcolemma. That allows for sodium to flood into the sarcoplasm, which generates an action potential to travel down into the T tubules. And then acetylcholine esterase cleans up acetylcholine and brings it back to the neuron. That is the full cycle of our neuromuscular junctions. So you should be very familiar with this process. This is a great thing to be prepared to be able to like explain hint, hint. So make sure you are able to explain what happens at a neuromuscular junction. And you should also be able to explain what happens to actually cause contraction, which we're going to get to in a couple slides here. So this is just a closer look at what a ligand-gated channel looks like. This is kind of an idea we'll expand more on, again, in our nervous system units or nervous system chapters. But this is just one version of a protein channel that allows for movement of things across a membrane. This is a ligand-gated channel, which means that it is binding with a ligand, some signal, like acetylcholine in this case, which is going to cause the change, the movement, in this case, the channel opening, so that sodium can enter the muscle cell. And I don't know if it is necessary to talk more about action potential in this chapter. We talk a lot about it in the next chapter. I think this has just been historically left here for context, but I think you can ignore the specifics of membrane potential changing. Again, this is just kind of exemplifying the idea that when ions move from one space to another, that changes the charge of the space. And that is what causes these electrical signals, this change in charge. It's basically all for here. Okay. So once we've wrapped up at the neuromuscular junction, right, an action potential has spread down the sarcolemma into those T tubules. And those T tubules end where they meet the sarcoplasmic reticulum, right? Those terminal cisterns. And those terminal cisterns are full of these calcium ions. So as the action potential reaches those terminal cisterns, that is going to cause the release of calcium, which, as I mentioned earlier, is super important for allowing muscle contraction. Its job is to bind with that troponin protein, which is going to cause it to change shape and move to the side, which pulls that rope-like tropomyosin out of the way. And so we see that exhibited here, right? Before calcium, that tropomyosin is blocking this yellow active site. So the magnet that wants to pull on these myosin heads, it can't do its job, so they cannot be attracted to each other. But when calcium arrives, that moves troponin, which moves tropomyosin. And now we have these exposed active sites, which myosin is immediately going to essentially grab onto, right? Like a magnet, boom. And that attachment between the thick and thin filaments is referred to as the cross bridge. Right? There is now a bridge that has been formed connecting the thick and the thin filaments. So cross bridge formation. And so here's another look. So now we're going to look through the entire process of contraction. So after calcium, or essentially zooming in on just the sarcomere, right? We've moved on from calcium release from the sarcoplasmic reticulum, right? And we're looking into the actual overlapping filaments and how they pull on each other. So in resting state, active site is blocked. Myosin head is in its cocked position, where it is holding on to this hydrolyzed ATP. ATP, as a reminder, has 3 phosphate groups. So if we pull one of those phosphates off, we're left with ADP, which is 2 phosphates. So in order for myosin to be in that cocked position, it essentially had to take an ATP molecule and pull one of those phosphates off, and that puts it into that position. So at rest, we are here with that cocked position with a hydrolyzed ATP. So calcium has been released, it bonds with troponin, which moves tropomyosin, and now that active site is exposed. So the head is going to be pulled onto it, forming that cross bridge. And now it will release that ADP and free phosphate, which is going to cause it to change shape and do that power stroke, what's called a power stroke, pulling the thin filaments closer to that M line towards the center of the sarcomere, which is shortening the entire sarcomere, causing that contraction. So the release of that ADP and free phosphate is what causes the power stroke after myosin has attached to the G actin. Okay. So now if we want to release that tension and go back to resting state, we have to add a new ATP molecule to each of these myosin heads. So they will stay attached. If there is no energy available, they will stay contracted. And so we need more energy to be added, which is going to cause these myosin heads to let go of that globular actin, but they are still in their contracted position. So in order to get back to resting state, they split that ATP, right? They pull that free phosphate off of it. So now we're back to ADP plus a free phosphate, and we're back in our COX position. And so now we can start over again. If calcium is still available and that active site is still exposed, myosin head is going to attach again, release ADP and free phosphate through the power stroke, and then we need more ATP to allow it to let go. But if the action potential has left already, the calcium will be cleaned up, tropomyosin will cover the active site again. And now to get our muscles back to their resting length, there needs to be some sort of opposing force. They don't necessarily snap back to resting length from contraction. They will stay overlapped unless some other force like gravity or an antagonistic muscle pulls them apart. The elasticity that we talked about before is going from an extended length back to their shortened resting state. But if they have been contracted, they will stay contracted unless some force pulls them apart. Okay? So keep that in mind. And that is essentially muscle contraction. So big ideas here, something we've already mentioned is, the calcium, right? I brought that up a few times now. Calcium is what moves these other proteins, allowing for the thin and thick filaments to interact. So that's the role of calcium here. New idea is that role of ATP. So we tend to think about energy usage and muscle contraction for causing the movement, for powering the contraction, which is true. We needed ATP held by these myosin heads in order to power that contraction. But you also need ATP to let go of that contraction. And so if you're having issues with energy production in your muscles or with ion concentration in your muscles, that is going to cause for sustained contraction, even when you're trying to release that tension and go back to resting state. And that's what causes a cramp, that like painful feeling of your muscles being stuck in that closed contracted states. That is that cramping feeling, that painful, ow, like I can't lengthen, I can't stretch out my leg because these myosin heads are stuck on the thin filament and they can't let go because we need to add energy to let go. This also explains the idea of rigor mortis, which I kind of jumped past here, where when an organism dies, it tends to, whatever muscles it had contracted are going to stay contracted because those cells are no longer producing energy to allow them to let go. So rigor mortis is describing the sustained muscle contraction in a dead organism, which is explained by this need for ATP. There are a couple slides here at the end, which I think do a really great job summarizing those steps. And so again, this entire process is something that you should be able to kind of explain in your own words. So how does a nervous signal get transmitted to a muscle cell at the neuromuscular junction, the NMJ? And then how does that nervous signal on the muscle cell end up leading to muscle contraction, right? How does calcium allow for the movement of these proteins on the thin filaments? And how does the myosin head attach to the thin filament and then pull it to the center? What is the role of energy and ATP in all of this? Right? Those are all concepts that you should expect to be able to explain in something like an exam. So that's where I will leave you for this portion of the chapter, and I will see you in the next one talking about tension and types of contractions.